next I would like to introduce our last-minute speaker, Dr. Alexey Soshnev. He completed a postdoc at Rockefeller University in New York with David Alice and started his lab at UT San Antonio last year in the Department of Neuroscience, Developmental and Regenerative Biology. His lab studies the role of chromatin in gene regulation, and he will tell us about his work on a linker, Histone mutant, identified a few years ago by Dr. Tatton Brown and colleagues in a related developmental disorder. Please welcome Dr. Soshnev to the stage.
Okay, thank you so much. And I apologize to the speaker whose slot I accidentally bumped. This was not my intention. This was a very impromptu presentation. I will try to be brief. I am a junior assistant professor at UTSA. I actually work in the same department as Jenny. And I started my lab about a year ago. And I have a project that I have been working on during my postdoc as a bit of a side project that I feel is very related to the current theme of this meeting. And so after about 2 days ago, then I learned about this this meeting. Basically, it was a bit of a scramble to come here and introduce myself. So today I will tell you about an unusual mutation in one of the linker histone genes that Dr. Tatton Brown knows well, that we think we now know how it works. So my interest in these circuits comes from the chromatin biology perspective, and chromatin doesn't really need much of an introduction to this crowd. I was doing my postdoc with Dave Alice at Rockefeller, where we studied mostly core histones, shown here. These are the protein complexes that DNA is wrapped around. They get modified by a number of enzymes, and then another class of proteins comes in and regulates transcriptional activity. And when I was in that lab, felt that the core histone field is quite crowded. And so I realized that there's one more histone protein shown here. It associates outside this nucleosome core particle. It's called linker histone H1. There are 10 genes that encode linker histones in human genome. They look like this. There's a structured globular domain in the center, and there's a very charged, positively charged C-terminal tail. Pretty much the only thing known about them is that they orchestrate higher order chromatin structure. So when I worked in that lab, we basically figured out that linker histones are very frequently mutated in human cancers, specifically in peripheral B-cell malignancies, which suggested that they have an interesting role in B-cell development. And these studies have been published. We characterized a number of linker histone mutations, and that made me a bit of an expert in linker histone mutational landscape. And what we realized at the point is that most of these mutations, absolute majority of these mutations, were actually single point mutations, missense mutations, that seem to be causing a loss of function mechanism in human malignancy. Again, these published studies connected linker histone function to two enzymatic complexes. Both of these were mentioned earlier in these talks. PRC2, which is a methyltransferase for lysine 27 on H3, and NSD1 and 2, which are methyltransferases for lysine 36. And then, of course, these are connected to DNMT3A. And just looking at this circuit, we got thinking about the developmental syndromes which are caused by germline mutations in PRC2 components, NSD and DNMT3A enzymes. And of course, at that point, we wondered whether H1 has any role in the circuit, and turns out that it does. And a number of patients that carry mutations in linker histone genes has been growing progressively. We started out with just a few that were characterized by Kate Tatton Brown. And then as the story progressed, we got in touch with a few other scientists who work on this. And now we have probably close to about 100 unrelated patients who all carry a very unusual type of mutation in this linker histone gene. Now, this is on a single linker histone gene. It's always a heterozygous mutation. And this heterozygous mutation always causes a frame shift into exact same frame that creates a de novo sequence, which varies anywhere between 75 and 50 amino acids. And structurally and biochemically looks quite different from the wild-type linker histone. Now that got us quite excited about this, so we went ahead and developed an antibody against this de novo sequence. This protein is accumulated in patient tissues. This protein is quite surprisingly localized to chromatin. So shown here is the immunofluorescence image of the wild type and a knock-in mouse AS cell line that stains not just the interphase nucleus for this frameshift protein, but also the metaphase chromosomes, suggesting that this protein stays in chromatin. And if we look at the transcriptional effects of expression of this frameshift protein, we see a very robust transcriptional signature. So we have hundreds of genes, depends on how you draw your cutoffs, that are now upregulated upon the expression of the frameshift protein. And thinking about this, we right away asked the question whether the effect of this mutation sequesters some partner off of chromatin, because that would be one possibility, or whether it brings something into chromatin and thus causes an ectopic transcriptional activation. So we used the same RNA sequencing pipeline to figure this out, essentially creating two additional constructs. One where this frame-shifted peptide was fused to another globular domain of a different linker histone, and another one where the frame-shifted peptide was introduced into the nucleus on the tail of GFP, which carries a nuclear localization sequence. And so what we find was pretty black and white, is that when the linker histone frame-shift peptide is in chromatin, it contains exact same ectopic transcriptional activity, but when we introduce it into the nucleus to let it freely float, it has absolutely no effect, which suggests that chromatin localization of this mutant peptide is absolutely essential for its activity. So then the question is, how does this protein function in chromatin? And of course, knowing the clinical structure of these syndromes, we first were looking for any connection to DNA methylation phenotypes, polycom activity, and activity of MSD1. And it turns out that if we look at K27 methylation and K36 methylation in this frameshift expressed cells, we have absolutely no effect. So this is some ChIP-seqs shown here. This is correlation plots showing that the mutant and wild type ChIP-seq dataset cluster absolutely together. There is pretty much no differences. Admittedly, we have not done full analysis on DNMT3A function, but just looking at DNA methylation profiles, we see no significant effect. However, when we performed attack sequencing on these frameshift cells, we see an interesting effect. We see that there is about 3,000 differentially accessible sites that are uniquely open in the frameshift expressing cells. This is always a unidirectional change. It's always increased in accessibility. These are very narrow regions that match a single transcription factor footprint. And these sites are actually occupied in the wild type and control cell lines, albeit at a lower level. Now again, as a sanity check, we'll look at whether these specifically attack-accessible sites have any change in K27 and K36 methylation, and we see none. However, when we map them to the genome, we see that these differentially accessible sites do localize next to genes involved in synapse organization, brain development, axonogenesis, which makes us quite excited about these. And when we look at the consensus motif underneath these differentially accessible sites, we find that all of them carry a very robust consensus site for a single transcription factor class, which is the basic leucine zipper transcription factors. Now, this suggests that possibly this frameshift peptide ectopically interacts with a basic leucine zipper transcription factor class. And if that's the case, we should be able to identify this interaction through a pull down. And so we express the wild type and frameshift linker histones in our favorite cells. We perform mass spectrometry to identify specific partners. And this is a volcano plot where the fold change is shown on the x-axis and the significance is shown on the y-axis. And of course, the most highly significant candidates would be in the top right corner. And it turns out that in the top right corner are 6 basic leucine zipper transcription factors that preferentially associate with this frameshift linker histone protein. We can identify the same interaction by a different assay using proximity ligation assay in cell culture, where each dot essentially corresponds to an interaction event using 2 different antibodies. And we can also quantify this interaction using genomics and available antibodies. Unfortunately, we don't have robust antibodies against all of these transcription factors. But in this case, we have one against MAFK. And we see increased accessibility, increased level of MAFK occupancy at differentially accessible attack sites, specifically in the frameshift mutant background. Now, this tells us that probably what's happening, at least in our culture model, is that in the wild-type situation, this transcription factor exhibits a certain dynamic class where it comes onto chromatin and then gets displaced off of chromatin. Whereas when the frameshift mutant is present, the frameshifted tail essentially provides a third leg for the transcription factor to associate with the chromatin, and thus locks it in its ectopic configuration and causes downstream transcriptional defects. And what I am very excited about for future directions of this project is to think more about how this is connected to the broader class of overgrowth and intellectual disability disorders, where we know that PRC2 and NSD1 mutations and DNMT3A mutations all operate in chromatin because these are paradigmatic chromatin factors. However, what exactly they do in chromatin, we still don't really know. And I'm quite excited to think about it in a sense that perhaps they misregulate the function of a certain transcription factor class that we might have identified using this linker histone frameshift mutation. And with that, I will give thanks to the lab that this got started in. This was at Rockefeller, and just recently I have my own lab at UT San Antonio. We are now funded by Cancer Society and NCI, and much of this project was initially funded by Star Cancer Consortium and Damon Runyon. And thank you very much, and I will take your questions.